Good evening, everyone. I'm Shweta Dhanda. So, before I start, I'm thoroughly pleased to witness you all here in this workshop. It's my privilege to address you all on the behalf of Dollar Education. I welcome all the participants in this workshop. We are truly delighted with your presence. We all have gathered here today to attend this workshop on research and career journey in bioinformatics. Our organization, Dollar Education, aim to achieve great heights. We are Dollar Education is an initiative to offer practical training on emerging concepts by making them affordable for all and developing a consistent environment of growing together in the field. What we want to promote bioinformatics and make everyone know the significance in the field of biology. The main concept is just to connect and share the ideas. You can also find us on social media with hashtag Dollar Education. What's in our name? We named our organization because it is a multifaceted meaning. It is a currency name in around 20 countries. It is a popular as an affordable symbol and also used as variable in computer languages. We want participants should get best of our workshop. I want you to be very interactive throughout the session so you can get best of it. Dollar Education also provide trusted information with proper references. We also provide meaningful networking. It could be a next career jump for you. We also appreciate diversity and multiculturalism. This workshop is something for everyone. For students, they can get better understanding of the fundamentals of this field. And for researchers, they can apply these ideas in their upcoming projects. Today, I'm pleased to welcome Dr. Bharat Panwar, Senior Scientist at Amigan, California, USA. He'll be sharing his experience and journey himself today. He completed his bachelor from J. Narayan Vyas University, Rajasthan, India, and Masters in Science from Biotechnology University of Rajasthan, India. Then he moved on Chandigarh and earned his PhD degree from a joint program of Institute of Microbial Technology, Chandigarh, and JNU, New Delhi. Next, he pursued interest and received postdoc training from University of Michigan and University of Immunology, USA. Currently, he is working as senior scientist at Amigan, California, USA. He is well recognized in the field of bioinformatics, immunology, and oncology. He has an outstanding track record of scientific publication. He also published cover page articles and also granted a US patent. He is also a well-known peer reviewer, mentor, and editor for many journals. Now, I welcome Dr. Panwar sir to start the session further. Sir.
Thank you so much, Sweta and Sandeep, for nice introduction. And let me share my screen. I hope all you can just. Yeah, can you see my screen now?
Yes, sir, it is visible.
Okay, so today's title of my talk is Data-Driven Drug Target Discovery. Since Dollar Education is interested in bioinformatics related seminars, so I'm keeping this seminar totally bioinformatics related. So thing is, when we normally do anything in the life sciences or biological sciences, we mostly focus on particular gene or particular protein. But now that we have so much advancement in the bioinformatics, so what we can achieve through totally hypothesis free and data driven approach, that will be my talk today. uh, so I'll start my today's talk the work I did at the La Jolla Institutes for Immunology, that's based on San Diego, California. So here I worked on two different projects. One project was the lupus, the systemic lupus erythematosus, that's the autoimmune disease, and that project was funded by a pharmaceutical company called Kyowa. And another project is immuno-oncology project, where we had the collaboration with the University of Southampton. But I'll talk about the lupus project today. Uh, so this was the institute where I worked before I joined the Amgen. And as you can see, the both Kyowa and La Jolla institutes, they were in the same building. So it was nicely collaborated project. What is lupus basically? This the it is very heterogeneous autoimmune disease. And autoimmune, when we say the autoimmune, where our own immune system start fighting against us, and it's called the great imitator. Great imitator means it always looks like something else, but it is a lupus disease. It can affect multiple organs, and it predominantly affect the women's. So multiple immune cell types, they are involved in the lupus. And here the B cells, normally they generate the autoantibodies against self antigens, and these autoantibodies then forms the immune complexes that can go and deposit into the different organs and then affect the normal functionality of those organs. So the main goal of this project was to, because it is very heterogeneous disease, it can affect multiple organs, and every person it is different. So that's why first our goal, can we find the molecular heterogeneity from the transcriptomic data or any kind of data. We predominantly use the transcriptomic data here, but first goal is to find the molecular heterogeneity. And second goal, once we find the molecular heterogeneity, then we have the clinical information as well, because we have the SLE cohort. So once we have the molecular heterogeneity, can we correlate that molecular heterogeneity with the clinical heterogeneity? And third goal was, because the multiple immune cell types, they are involved in this disease, how can we understand the crosstalk between the immune cells using this transcriptomic data? And the final goal, because the project was funded by Kyowa Pharmaceuticals, and they were interested in identifying the novel therapeutic targets in the SLE pathogenesis. So this was the SLE cohort. Uh, we had the 24 healthy control and 64 lupus subjects or patients. And they all women. As you can see, there were only one man there, because it mostly affect the women's. And we have the all these demographic informations and different clinical features, like what kind of treatment they were taking, or the different cytokines levels. And we had the transcriptomic data, that's the bulk transcriptomic data, but still we have these for purified different immune cell types. For 6 different immune cell types, we have the data: B cells, T cells, and 2 different type of dendritic cells, neutrophils, and the classical monocytes. So for the classical monocyte, we had the data for all cohort, and for other 5 immune cell types, we had the data for subset of the cohort. So first we started our analysis with the classical monocytes, because this was very well known in the lupus, and also because we had the data from most of the cohort. So first we simply performed the differential gene expression analysis between the lupus and healthy control. And what we really find is these 125 differentially expressed genes when we compared 64 lupus patient with the 24 healthy control. And when we did the pathway enrichment analysis for these genes, we found though most of these genes, they are related to interferons somewhere. So we have selected 20 uh, very well known interferon signature genes. And when we looked into the expression of these genes As you can see in the healthy control, the green here, I'll use the same color scheme all over my presentation. So green here is the healthy control, 24 subjects. It seems like these interferon signature genes, they don't have expression. And then half of the subjects, the interferon positive subjects, 30 out of these 64, 30 subjects, they have very high expression of these genes. Whereas 34, those are interferon negative, we call them interferon negative, they don't have expression at all. It means that there's two different groups of the lupus patients. They're interferon positive and interferon negative. Almost half. So half of the subjects, they have the expression of the interferon signature genes, and half of the subjects, they don't have expression. It means like clearly there are two molecular subgroups present in the lupus disease based on the interferon signatures. And now if you see the same PCA plot here, so the green healthy control, they are very close to blue here, that's interferon negative. But if you see the interferon positive subjects, they are very different from there. And now that we have these groups, we wanted to know whether these interferon uh, expression, whether it comes from the multiple cell types, multiple cells, or it comes from only fraction of the classical monocyte cells. So we did around like single cells sequencing for 150 single cells from the three different interferon positive subjects, and as you can see, most of the cells, they produce some kind of expressions. It means like the interferon produced by most of the single classical monocyte cells. And these are the two important interferon related genes that you can see the expression comes from all the cells. And after that, we look into the protein level expression as well. We had the cytokines data. Interferon alpha looks different, and the interferon beta was also different, but interferon gamma wasn't significantly different between these two. And we look into some other cytokines as well, and these two pro-inflammatory cytokines, interleukin 6 and the IP10, that we find significantly different between interferon positives and negatives. So not only at the transcriptomic or RNA level expression, we look into the protein level expression as well, and we find the difference there. And after that, we looked into the longitudinal data as well, because in the cohort, some of the subjects, we had the longitudinal data. So as you can see here, these are all the subjects, and then their visit number. For example, this subject S1055 had the 6 different visits, and the color, so the red color here for interferon positive, this subject was interferon positive based on those 20 interferon signature genes what we used in the classifications, and it remained interferon positive in all consecutive 6 visits. So most of the subjects, they remain in the same categories, whether it was interferon positive or negative, except one subject, this one S1057. In first visit, it was interferon positive, then it turned into the interferon negative and remained interferon negative in following 3 visits. So when we look into the expression of these IP10 and the CXCL10, so IP10 basically the same genes, it's the protein level, and then this is the transcript level. And as you can see, when it was interferon positive as first visit, the level was higher in both protein and RNA level, but in the following visits, the expression goes down for both the RNA and protein level. And similar with the interferon gamma, you can see, and the interleukin 6, the same pattern there. So thing is, like, so far what we learned is simply we compare the healthy control with the lupus patient, and we find the interferon related pathway, and based on that we selected 20 interferon genes and classified lupus patient into interferon positive and negative. So here we did all based on the differential expression analysis, but in the differential expression analysis we treat at each gene as separately. But in the biological system, you see the set of genes, they comes together and then they perform particular biological functions. And since we have the transcriptomic data here, we wanted to take leverage of that data, and so to understand actually what's going on at the system level. So one of the tool that's very classic tool in the bioinformatics, WGCNA, that's the weighted gene co-expression network analysis. Here you can take your whole transcriptome profile, whether it's come from the RNA-seq or microarray, and then you can generate the network based on the co-expression. And once you have the network, then you can generate the modules or cluster. So each module is set of genes, they co-expressed with each other. And once you have these modules, Then you can generate the eigengenes or the PC1 values for each module, and that you can correlate with the external any external clinical features you have, for example age, sex, or any other information you can correlate. And then you can basically link the modules. Those are like set of co-expressed genes that is linking with your clinical features. So that's the beauty of this tool. Basically you can link any clinical features with your molecular features, molecular modules that you have. And also you can generate two different co-expression network. For example, here we know that there's two different set of uh, SLE patients, they're interferon positive and interferon negative. So we can generate the separate network for interferon positive and interferon negative, and then we can compare these networks as well. And once we find any interesting module, then we always have the gene level resolution. So basically we can go back and find within that module what are the hub genes there. So this is the overall strategy that how you can use the WGCNA. And so first, when we so here we use all the lupus data from 64%, and we develop the one co-expression network. And as you can see here, these are all the modules, and in the bracket the number of genes present in these modules. And so it's not like for every gene you find the co-express partner. If you don't find the co-express partner, then they put those genes into the gray modules. Gray modules mean these genes, around 5,000 genes, they don't have any co-expressed gene in the network. But in the differential expression analysis, you choose one particular comparison, for example, positive and negative, then you do the comparisons. But in the WGCNA, it's not, you can have any number of the clinical features here. For example, the interferon uh, class, whether it's interferon positive or negative, that we used here and correlate with the eigengenes or PC1 for each of these modules. And then we use different clinical features like age, or all demographic informations, or the flare severity, SLEDAI, that's the SLE disease activity index, like how severe this disease is. Based on that, clinician they have these scores, and years of disease, or whether they are taking any kind of medicines or immunosuppressors. So basically you can correlate with any clinical features. And as you can see here, when we did this correlation, we did not find any of these significant correlation here except the interferon one. So it means like interferon that is really very strongly correlate, and very strong signal available present in this lupus actually data. And what we find basically is this blue module, as you can see, is very strongly correlated, around 0.83. The correlation is strongly correlated with this particular module, the blue. And then there's another module, the red, is the negatively correlated. And these are the genes based in the blue module. And then earlier we used the 20 interferon signature genes, but later we extended our interferon database. So we include all the indirectly genes, those are indirectly related to interferon. There are around 363 genes. And as you can see, they are all here in the purple color. So a lot of interferon related genes, they are there in the blue modules, although there are some of the genes they are not related to interferon as well, but most of the genes coming from the interferon, somewhere related to interferon pathways. And now next, how we can compare these two networks. We generated the why it is important first, why it is important to compare the network. So as you can say, like, because we have the two different class, interferon positive and interferon negative, you generate the separate network and then you compare. But why it is important? For example, if you have any transcription factor in the interferon positive, and that has the expression average action, for example, see the TPM, that's the transcript per million of 10. And in the interferon negative also, the same transcription factor have almost similar expression. You won't find this transcription factor as a differentially expressed, but the different cofactor or the other genes, there, how many co-expressed with this transcription factor, they can be different. So overall, how this transcription factor connected in that network, that can be different. So we can find the differentially connected genes as well. So overall goal was to find the potential therapeutic target, because we start with 20,000 or something genes from totally unbiased data driven. But how can we narrow down and find out at least like 5 or 10 those are the potential target that the queue occur in and they can try. So that was the whole goal. So once we find these two groups and learn what are the differential express genes, further goal how we can further narrow their list down. So here what we did, we generated this separate network, interferon positive and negative, and each gene we compared their connectivity. So what you see here in this plot, x-axis, that's the difference in connectivity. So all and each dot here is one gene, and the color based on in the interferon positive pathways based on their module color. So all the genes here in the right hand side, they are they have the higher connectivity in the interferon positive pathway. It means they have the higher number of genes connected in the interferon positive pathway in comparison to interferon negative pathway. And these are the genes in the left hand side. They have the higher connectivity in the interferon negative pathway. And in the y-axis, this in the DSC2 based the fold change. So it means all the higher they have the higher expression in interferon positives in compared to interferon negative. So what is really important is this box. We were interested in this box only. So all these genes here, they are more connected in the interferon positive subjects, as well as they have the more expression in the interferon positive subjects. And so when we use this criteria, uh, significantly differentially expressed as well as significantly differentially connected, we find around 64 genes. And in those 64 genes, you can see in the red here, they are related to interferons. There are some genes, they are not related to interferon. And two very interesting genes, what we find is the TNF SF13D. That's what we call BAF as well, B cell activation factor. And then IL-1RN, that's the interleukin 1 antagonist. So these two genes, they are already known drug targets. Uh, so this BAF, B cell activation factor, is a cytokine of the TNF ligand family, and it's a potent B cell activator. Uh, so as we know, because this is autoimmune disease, and the B cells, they generate a lot of autoantibodies. So whole goal here, how we can target these B cells so that basically we get the less autoantibodies. And GSK, they developed this, they already marketed this belimumab. So that is based on the BAF, it targets the BAF so that you can control the autoantibodies. And once we find that this was in the central genes, what we narrowed down, so then it's kind of built some of our confidence. And the another interleukin 1 antagonist, that's basically interleukin 1 signal. Normally, when it binds to the interleukin 1 receptors, it gives the all the pro-inflammatory signals. So interleukin when antagonist basically when it binds, it stop all those inflammatory signals. So this is anti-inflammatory. And the Amgen, they have this drug anakinra. That's basically the recombinant form of this interleukin 1 antagonist, and they have this for the rheumatoid arthritis. So as you can see, the uh, both of these anti-BAF and these uh, interleukin 1 antagonists, they were already known drug targets for the in the autoimmune space. So that was the, and these comes from the totally data driven unbiased analysis. So it was really built our confidence. And then we find this interleukin antagonist, they several reports there that they used in the lupus as well, they find this useful. So now you see here is the same plot what I have showed you earlier. And the gray, if you compare with the gray, as I told you that gray, all these grays, they are the genes where they don't have any co-expressed partner. And then these the green module, what we find, most of those 64 genes, they are all come from the one particular module, that green module. See like how they are very different. So all the genes in the gray modules, they are more connected and more expressed in the interferon positive subjects, and also because they are co-expressed with each other, that's why they are in the one green module. And if you talk about these two BAF and the interleukin 1 antagonist, as you can see, if you see in the network with the AS weight higher than 0.05, 0.05, you don't find a single gene that's basically connected with these two in the interferon negative pathway. But if you compare in the interferon positive pathway, see, a lot of genes they connect with these two. So that clearly shows that these two already non-drug targets, they basically have lot of uh, connected genes in interferon positive pathway. It means like it is important there somewhere biologically. And so once you prescribe the drug, you should take care of this information also into consideration, whether it's interferon positive or interferon negative subjects. And these two correlate very well with each other. And uh, so SLEDAI scores I talked about earlier, that's the SLE disease activity index. And in this paper, they classify these scores because this range from 0 to 24 based on how many organs are affected or how severe is this disease. So physicians basically they give these scores. And as you can see, the disease activity index one they classified into three different class. When the score is 0 to 2, or when score 3 to 7, it's kind of moderate disease activity 2, and when it says 8 or higher, then it's the disease activity 3. And so if you see the expression of these uh, two genes based on this classification, so healthy control and these three class in the interferon negative, not much difference. But in the interferon positive, the expression of this gene increase with the increasing SLEDAI grade. So it shows that the expression of both of these genes also correlated with this how severe the disease is. And in one particular subject, if you remember, where we see the switch of the interferon status from positive to negative, there also it goes down, expression of both BAF and the interleukin 1 antagonist. So uh, what we learned so far is that we find two different groups from differential gene expression analysis. Then we did uh, differential network analysis, and we further find narrow down to 64 genes. Out of those 64, two already non-drug targets. So what else we can learn from here? The next, because so far whatever we did is from the classical monocytes, and now we have the subset of the data from 5 different immune cell types as well. Uh, so we wanted to see whether this interferon signal, what we're seeing from the classical monocytes, whether this is consistent in different immune cell types or not. So first, if you just simply see based on the most variable genes, for example, the 1,000 most variable genes, this is how it looks like. All the immune cell types, they cluster different. But if you do this clustering only based on those 24 interferon signature genes, then this is how it looks like. And if you just color based on these status that we derived from in the classical monocytes, then you can see all these interferon positive here. So it means this signature is very dominating, and this is cell type independent. It means the same subjects, they were the interferon positive and negative across different immune cell types, as you can see here in the B cells or neutrophils, T cells, plasmacytoid dendritic cells, or classical dendritic cells and monocytes. So all these immune cell types, uh, interferon positive and interferon negative signature is still intact there. So up, and also you can see at the gene level expression as well, for the expression of these interferon signature genes in the monocytes, T cells, and red here the interferon positive subjects. You don't see expression in the interferon negative or healthy control. There's little here in one subject that was infected from some other, actually some he had some other viral infection, that's why you see little here. But mostly there's no expression in healthy control and the interferon negative. They all expressed in the interferon positive, and this is kind of average of those uh, 20 signature genes in different monocytes, sorry, different cell types. And if you see the expression of these BAF and the interleukin 1 antagonist, although we find this in the classical monocytes, but the expression is very high in the neutrophils. And but still you see in the monocytes and neutrophils also the expression higher in the interferon positive subjects. So Then we perform the different differentially gene expression analysis for all different cell types. First, we did for the we already did for the classical monocytes, but for all other 5 immune cell types also we did these comparisons, lupus versus healthy control. And as you can see, when you compare the interferon positive with healthy control, you see the more higher number of differentially expressed genes. But interferon positive, sorry, the interferon negative versus healthy control, you don't see that many genes differentially expressed. It means like their transcriptome profile is almost similar. And the positive versus interferon negative, we see a differentially expressed genes there. And these are the overlapping genes. There are some genes, they are uh, exclusively differentially expressed in that particular cell type. Some genes, they are overlapping. These 35 genes, mostly they are related to interferon. But thing is, once you have the all different kind of differential gene expression comparisons, how to systematically analyze that, how to learn that these immune cell types, how they interact with each other. So that's why we come up with this method. So in the normal WGCNA, as we learned that you can build a co-expression network, but standard WGCNA, you don't have functionality to basically learn about how the different immune cell types, they can crosstalk with each other. So we come up with this new method, that the multi-cell WGCNA or MWGCNA. So you can basically use all the transcriptome profile from different immune cell types together and generate one big network. And then you further cluster it and do the all the analysis. So let me show you how you can do this. So for example, this is the transcriptomes from different immune cell types, from B cells, T cells. Each row that you can see is basically the transcript there. uh, so you merge all this data together, but still you keep the identity that this comes from the B cells or T cells or dendritic cells, and then generate one network, and then you find the different modules. So now in the each module you can have the genes that come from the B cells or come from the T cells. So basically this is how you generate the modules. So we remove the low expressed one genes, and then around from the 6 different immune cell types, we had total around 100, higher than 100,000 total genes. And then we generate this network and uh, generate the 78 different modules. So as you can see here, in each module, genes can come from different cell types. So for example, in this, the largest module, most of these cells come from the B cells, but its fraction can be different, the proportion can change, but at least we see that from different immune cell types, genes can come together. So why it is important? For example, some of the genes like biological function from the housekeeping genes, or they are very constant. So they, hello, yeah. So uh, but what we, yeah, so most of the biological processes, they are very constant, that we can see here, like they, all the genes from different immune cell types, they will show some kind of co-expression and will come into the one particular module. But whenever there is some kind of the changes, we can observe from here. So, and another thing, that we can always correlate this with the clinical features we have. So what we actually did here, instead of correlating with the clinical features, we use the differentially expressed genes from all different immune cell types when we compare their interferon positive and interferon negative. So for example, we did this comparison from the B cells and from the T cells, from simple differentially expressed genes, and then we looked into the enrichment, which module we see the enrichment of these differentially expressed genes from B cells or from T cells. That's based on the hypergeometric test, the how you calculate the enrichment. And so as you can see, some of the modules we see the enrichment of the differentially expressed genes from the PMN or the neutrophils or from the classical monocytes or dendritic cells. But one particular module here, black module, that's we see the enrichment from the all 6 different immune cell types. It is very consistent. So that we wanted to further explore that. And when we correlate with the clinical features also, that shows the highest correlation with the interferon. And then second is the severity. And when we look into these black modules, we find they mostly correlated, mostly enriched with the interferon related genes that we already had the database of 363 genes. When we looked into most of the genes, they are related to interferons, and these BAF actually the genes from 363 different immune cells, monocytes, neutrophil, and dendritic cells was in the black module. And similarly, this interleukin 1 antagonist from two different immune cell types was in these modules. So that basically reconfirmed what we initially find, that these are the important related to interferon, and these are important. So this is the overall uh, pipeline what we come up with finally, that our strategy to prioritize the potential target against the lupus for the all immune cell types we used one by one. So first we did the differential expressed analysis for the each immune cell type separately, and then we generated the differential connected differentially differential network analysis and find out the differentially connected genes between the interferon positive and negatives, and then we look into their intersections, and also we remove the uh, genes related to interferon, because the Kyovakirin, they were say, although the interferon is dominate very dominating signature, but they wanted to explore the genes those are not related directly related to interferon. So we kind of use the non-interferon related genes, and then we looked into some expression as well, picked the highly expressed genes, and then for all the genes we picked up here, we look into the correlations with the SLEDAI score, that's the disease activity index score, and then we also looked into if there's any non-EQTLs from the in-house database, and then we will further verify with the multi-cell WGCNA and from the public cohorts as well. And finally, we come up with the prioritized gene list of like 12 genes that are being investigated now in the Kyovakirin. So this was the overall strategy for the lupus. But why I presented this like story, because I think this is totally bioinformatics story, totally data driven story, and where you can finally see some kind of clinical relevance. And so I think the same strategy you can apply for any kind of analysis you are doing where you have some kind of this transcriptomic data, so you can adopt this strategy. And this was the summary of the our lupus project. The interferon signal is the most prominent signatures that leads to the large gene expression differences between the positives and negatives. So based on the differential expression analysis, we find these two molecular groups that then also we look into the correlations with the clinical features. And to narrow down the list, we did the differential network analysis as well, and we find the 64 genes from out of that, two already known drug targets there. And we find these two important genes, they are highly connected in the interferon positive network, not in the interferon negative network. And the multi-cell WGCN analysis that reconfirm what molecules we find earlier and suggest they are the potential new targets. And what we learn is that studying the different immune cell types, both the individually and jointly, that is important for lupus drug discovery, but I think like for any other kind of the drug discovery as well. And so we published this research as a cover story of the genome research. And so this art basically the Anne Kim Anne Kirnan, she's a artist based on the Germany. She generated this art where she showed this butterfly, as you can see. So butterfly shaped signature, that is the signatures for the lupus disease. And she showed this butterfly in two different colors. That's basically the interferon positive or negative two molecular subgroups what we find. So yeah, that's the story, and I welcome any of the question you have.
Thank you, Dr. Pawar. Now we encourage participants to kind of ask question interactively so they can unmute themselves and they can ask questions.
Hi, Bharat. It was a very, very wonderful talk. Like it was really wonderful. I have a couple of questions, but I'll just ask few and let other participants ask, and then maybe I can. Is it okay if I can contact you and ask multiple questions?
Yeah, sure. That's definitely
Okay.
fine. Yeah.
So first one is, like you said, you used RNA-seq data, single cell data
Yes.
for that
Yes.
cohort and subset of that cohort. But I remember one of the slide you showed protein expression also. I
Yes.
was wondering, where does it come from?
Yeah. So the because the whole cohort generated here in the based on San Diego, we have the Script Institute as well as the UCSD hospital, and this was sponsored by the QR Caring. So they generated these transcriptomic data, but they also had some of these protein expression data. I think they generated through some of their vendors, the local vendors.
Okay.
Yeah.
That's great. And my other question is, MWGCNA, is it already out there? Like our package or GitHub is already out there?
Yeah. So as you can see, the when we say the MWG CNA, let me go to that slide. It's just the strategy. So you can simply you already have the WGSN. It's just
Yeah.
the how can you use that. But the differential analysis part, if you go to, there, we already have the code in the GitHub. So like how we did the differential part, like how you can generate the separate network for positive and negative and then compare that. And for MWGCN also, all you need to basically combine the transcriptome profile. But thing is like how you can learn, like once you have these modules, you can correlate with the clinical features. But I have the like we did some other analysis also that you can perform. For example, if you find any of the modules interesting, then here further you can see how many genes coming from, for example, cell types 1 or cell types 2, and then you can generate the separate eigen genes or PC1 values for each cell type and then correlate with each other. So that's how totally unbiased way you can find which of the modules you see the crosstalk. That we did actually for another project, for immuno-oncology project that I'm not showing here, but the paper that's in the bioarchive available, you can see there. So
Do you have a
uh,
link for that, or like the topic?
yeah, sure. Yeah, so that's
Okay.
so. For example, these two different cell types, CD8 and CD4 cell types, uh, you have these two transcriptome, and then you can merge and generate the modules. And now from the each module, you can basically either correlate with any clinical features you have outside, or you can generate these eigen genes. For example, here you can generate the eigen genes from these genes separately, and then from turquoise and yellow separately, and then correlate with each other. So you can do same for the all these modules and find out which module you see the more crosstalk.
Okay. And integrative, when you say integrative WGCNA, it's integrative of two data. Like, do you use any tools for that, or it's just integrative means like combining two data, like literally combining two data?
Yeah, just combine like two data, and you can use, for example, any you can use as a suffix, like for example CD4 and CD8. Yeah, in that gene, so you know that it's coming from CD4 or CD8 or any cell types you have.
Okay, great.
Yeah.
Thank you so much.
Okay. Yeah, so in the GitHub, the code is available. You can directly use that. It's just like how you can twist that based on the your project requirement. Uh, because to overall, like the I think the WGSN is very well written and very like it's from
Yeah.
2006 or 7.
Yeah, I use WGCNA a lot, but
Mhm.
with clinical data also. But
Mhm.
like you have in-house data, right? So, but I use like publicly available data, so it's not nicely. The cohort is not that nicely organized. Like the same
Mhm.
I don't get the same cohort with for RNA-seq or single cell, or so the integration is kind of uh, where
Mhm.
I struggle. It's like I don't have the same data.
Okay, okay. Yeah. So what, like you use the public data?
Yes.
Okay. So from like one of the thing, for example, if you, even from the public data, if you have the, if you know the molecular groups and you generate the separate network, then I think in the, they already have the tutorial where you can see, for example, if you find any of the modules, or like the, for example, 100 genes, the one module of 100 genes in one particular network, network one, then you can see whether that module remain intact or it kind of get destroyed
Mhm.
in the other network. So that's how you can rank the modules. For example, okay, these set of genes, they are co-expressing in the network one, but they were totally random in the network two, like they are going into the gray module. So that's those genes are important basically, because they are kind of somewhere, uh, they provide the more information than the differential expression, because they are co-expressing also in only one particular molecular subgroup. So there must be something biologically relevance there.
Mhm. Do you also see like multiple modules which are highly correlated? Let's say, like you have green and gray.
Mhm.
Like gray is, of course, the genes which are not annotated nicely or studied nicely. But like, for example, what if you might have got green, blue, and red with, yes, like 2, 3 modules which are highly correlated with IFN.
Yeah. So as you can see here, uh, here in the right hand side, they all differentially, they have the more connectivity in the interferon positives. And if you see in that box, mostly they are in the green modules. They're all in the green.
Mm-hmm.
So we didn't find any other modules. For example, turquoise you see here, they are differentially connected turquoise, because turquoise normally they in the largest
Yeah.
module they just give the
Yeah.
color means nothing. It's just like to identify that they co-express together. But they are the differentially connected, but there was not difference in the expression. So we wanted to like use this where they have the both, actually they are differentially expressed as well as the differentially connected. So that's why we use this. Yeah, that's why we use actually this part here.
And this is D-seq, like DG, differentially expressed genes, right? But in WGCNA, we don't use differentially expressed genes. We use the raw data.
Yeah, so this is separately. So for
Oh.
each gene, you calculate the difference in connectivity, and from DSIG, you already have like their fold chains.
Mm-hmm.
So you can use like simple t-test as well based on these.
Mm-hmm. Okay.
But yeah, because we already did the differential analysis, so I used actually their fold chains from DSIG.
Great. Thank you so much. Thank you so much. I might ask you a couple of more questions, maybe.
Yeah, sure.
Yeah. Okay. Thank you.
A great talk. Thank you so much. Can you hear me?
Hi, Sunil. Yes, I can
Yeah,
hear.
so yeah, it was a great talk. Uh,
You.
I don't have much knowledge about this subject as such, but I have a few queries that I would like to address
Yes,
to
sure.
you. SLE is a genetic disease as well, because I read that these Native North Americans and African Americans and Asians, they are more susceptible to this. So has there any genetic study been done as to what exactly is the gene responsible, or SNP is discovered or something of that kind? Please pardon me for my ignorance because
No,
I do
yeah,
not know much about it.
that's really a great question. So some of these genes like they find in the genetic studies, but overall you see the even like in the different epidemiologies, like they find, for example, the African Americans, they have the higher chances.
Hmm.
And but still I think we know very little at the molecular level, because it's very heterogeneous. You see,
Hmm.
it's like 20 plus different diseases, we call them lupus.
Okay.
Yeah, it's very heterogeneous. That's why it's, you always find something, but hard to make kind of that the applicable for all the lupus patients. So
Right.
heterogeneity is the major problem. I think this is the most heterogeneous disease, lupus.
Okay. Okay.
Yeah.
And the other question is, how are we placed right now with the treatment of this disease? Because I was reading it's only symptomatic treatment that we do. like giving steroids and
Yeah, mostly
bringing
these
down the
immunosuppressants.
inflammation.
Yes,
And
that's correct.
SLE as per doesn't cause the death, but due to the inflammatory disorders and things of those kinds, that can lead to the death. So are we just grappling with these inflammatory reactions, or we have something tangible that we can
Mhm.
address the disease itself per se?
Yeah, so this uh, anti-BAF basically that is used for the lupus, but mostly it managed by the immunosuppressors. And
Mhm.
since it is not that severe, it's just like sometime it get and sometimes it can get very severe, and also because it depends on what organ got like the infected. Like lupus nephritis, it can get really very serious.
Mhm.
Yeah, but mostly it's like this is so overall people manage with these immunosuppressors. There shouldn't much problem there. And but if you see the targeted drug, I think this is the only drug, the anti-buff we have. And the
Mhm.
recently the AstraZeneca, they also have the drug now that basically target the interferon receptor.
Okay.
So yeah, we have like some of the drugs, uh, especially in the clinical like the drug development space. This considered as a kind of graveyard of failure, the lupus, because it's
Mhm.
very hard to find something from here because of the heterogeneity. But yeah,
Okay.
day by day through all this data we are learning more. So yeah,
So
that
basically,
answer.
what promising drugs we have are mainly the monoclonal antibodies, is it? That we try to
Yes, basically
target
the
target
belimumab.
those.
Yes, yeah,
Yeah. Okay.
belimumab, or like you
Okay. It was
target the interferon receptor.
okay. Thank you very much. I would like to learn more about this because it seems to be very very interesting. I read about SLE in my
Mhm.
college days, but
Okay, great. Yeah,
thank you
thanks
so
for your
much.
interest. Yeah.
Thank you. Thank you.
There is one question from the chat that I would like to read. Poonam is asking, sir, can you elaborate on how lifestyle affects onset of autoimmune inflammatory response and molecular pathology?
The lifestyle.
Yeah.
Yeah, so that's really like, so the environmental factor, because people get, for example, the allergies and like from the different, for example, from pollens where you live, and so that definitely affect your autoimmunity. But overall, yeah, overall, if you see the lifestyle wise, so it's I really wanted to present the other project as well, immuno-oncology, because these two projects, they were totally different, but they are still the same. Because the lupus, that's the autoimmune disease where your own immune system work against you. So it's kind of your immune system is hyperactive, and whole goal is how you can suppress your immune system through these immunosuppressors or like just suppress your immune system. But in immune oncology, it's the opposite. Your immune system is already so suppressed. You want to activate that so that can fight against the cancer. But so now through the lifestyle, you see like how come your immune system become hyperactive. What can be the factor that uh, that contribute into like hyperactive immune system? Maybe like some diets, like some of the food you are eating, they are related to that. So definitely, or like yeah, like whether you have the balanced immune system or not, like through the exercise, or definitely that affects like both side, because whether it's very suppressed or hyperactive, both side it is not good. You need kind of balanced immune system, overall immune system, balanced immune system, and that uh, your lifestyle directly contributing to that. There's so many studies around that.
So can I ask one more question? Like, since you
Mhm.
have worked with immuno-oncology also,
Yes.
the Treg population, how does it compare in the two cancer and this particular inflammatory, because I believe the Treg population has a major role in both of them,
Yeah,
inflammatory as well
definitely.
as
Yeah,
cancer.
yeah,
So,
that's really good. That is great. Yeah. Do you have any other question?
so how is the population varying in both the diseases? Like, I
Yes,
mean,
yes, definitely. So T regulatory cells. So here in the, for example, if you understand in case of the lupus, where T
yeah,
regulatory cells they suppress the immune system, that's really good.
right.
You want to have the higher number of T regulatory cells in the
Hmm.
case of lupus, but it's just opposite in the immune oncology. If
Immune-
you see the higher number of T regulatory cells, that's bad. We want
Yeah.
to basically target that. We want the less and less Treg cells. And also the Treg cells, for example, we define based on some of the markers like FOXP3 marker or something.
Hmm.
But now all these, the advancement we see even like in the Treg cells, for example, the T follicular regulatory cells, the
Hmm.
TFR cells, they are
Yeah.
even, they have the more suppressive power in
Hmm.
compared to the normal classic T regulatory cells. So we are learning more and more actually about the different immune cell types. But definitely, like, see, I think you pointed really very right. Actually, the immune cells that has totally different, like how we want, how we think about that immune cells
Hmm.
when we work on the autoimmunity or in the immune oncology. It's just
So
opposite.
you worked on both the sides,
Both the
a
sides. Yeah.
hyperactive and very suppressive environment.
Yeah, I think that that was really great learning, like to look into both sides and
Hmm.
at the same time. So that's interesting.
Thank you.
Yes. Is there any other question? Yeah, no, you can always actually reach out to me on LinkedIn if you have any other question. I'll be happy to answer.
I have another question if there is no
Mhm.
other question.
Yeah, sure.
I just want to know your thoughts on the same WGCNA, but your thoughts on like interspecies, like WGCNA is a network correlation of genes and clinical
Mhm.
outcomes or events, right?
Yes.
So, like I said, I don't have the available data for the cohort human data,
Mhm.
but when I look into the data, I have like mouse data for something that I'm studying and then some
Mhm.
human, some. So, what do you think about the interspecies WGC, like human and mouse together, and
Mhm.
like there is an interspecies WGC? What do you like?
Yeah,
I want to know your thoughts on that.
yeah, definitely you can do that. So you can have one network. So only thing is like when you do the comparisons, you need to first keep the same overlapping genes that you can use. So make sure all the gene symbols you use, they are the they're actually tools to use that. So you can have the similar gene symbols from both the human and the mice, and then you generate the network for the human separately and the mice separately, and then you can compare networks. So then you can find actually some of these modules like they are different. So you can say, oh, these biological processes they are very different from the human and the mice. You can, yeah, definitely you can do that analysis. Uh,
Okay.
So basically, you can compare any of the two groups that you have, but only requirement is that you need at least 20 plus samples in one particular network.
Yeah.
So when you generate the network, that is the recommended size. Uh, and but if you don't have like the cohort for your own, but I think there's plenty of data available now. If you go to that genomic data commons, they all have the data from TCGA. They have all kind of data, the transcriptomics, proteomics, or the mutations data, copy number variation, everything. So I don't think there's any limitation like data, like you have to have generate your own data.
Yeah, that's true. But so I work in human organ transplantation, particularly kidney and liver, and
Mhm.
there actually is like there are data, but like for single cell kind of data, there is very limited data, and of course it's like for human samples, or it's very limited that
Mhm.
uh, like maybe 3 years back, and where they are using tools which are
Mhm.
not that great now. It's like so whatever the outcome or whatever they are concluding in the
Mhm.
paper now it's not even relevant because the data how they analyze is very different and
Mhm.
it's not that
Yeah.
great. So
So I
that is the problem with the availability
Even
for.
in the single cell data, there are a lot of data publicly available. And
Mhm.
so I feel the smart bioinformaticians really don't need their own data.
Yeah, that's
Yeah,
true.
they can always find the data publicly available where other people, they kind of
Yeah.
uh, spend a lot of money and generated data for published paper, but all the data available there for your
You just
analysis.
need a paper in
Yeah,
bioarchive and like looking out for, oh, this is the paper that's coming, and that's it.
yeah.
That's
So
true. Okay, great.
definitely.
Thank you.
Yes. So should we directly continue to the career journey, or people want to take some break? Uh,
I think let's continue to the career journey if you don't want to have break by yourself.
uh, okay. So I think let me take actually 2 minute break. Then I'll
Yeah,
be back.
sure. So since Dr. Bawar is on break for 2 minutes, we can use this time to get some feedback from the participants. Are they liking this kind of journeys, or what's their thought on this? So we wanted to make it as interactive as possible.
So I feel, Sandeep, this is a very good effort that you people are doing, and you're bringing together many good speakers, and this will be very motivating for students, particularly. Like they can identify what exactly are the very major thrust areas where they can make a contribution, and to learn from the people who have already had this journey. This is, I think, a very good way to know what are the hurdles and what are the challenges and how you can go about solving them. So I congratulate you and other people in the dollar education that you are bringing out this series. This is a very novel idea, and this is something away from the normal science lectures that we have. Because
Thank you, sir.
yeah, I think this is very good, Sandeep. Thank you so much. And I
Thank,
encourage all my students to join this group and have a experience from the people who have already undertaken such journey. And
yeah. And especially like I believe like field like bioinformatics has like very fertile ground in under
yes, yes,
under resources countries
right. And
because
your journey is very inspiring. So I
thank you, sir.
always quote my students that you should learn about Sandeep, connect with him, and we are really proud of you and the work that you are doing. Thank
Thank
you.
you, sir. Appreciate your kind words. Any other feedback that you like to speak on before we move on to career journey by Dr. Pamar?
Am I audible?
Yes, you are.
Yes.
Yeah. I just want to say that as a student, it's so informative and inspirational to learn about the journeys of the such a brilliant scientist and the faculty members, and to know the hardships they have faced and learn how to overcome the obstacles is really that inspired us a lot. And particularly as far as myself is concerned, I am very much interested to learn all these things. And because as a biology students, we are generally focused on the classroom teaching and all that, and we generally miss all these kind of things that has been practically applicable to the all this process and the journey of this from a student to a researcher and then to a scientist. So it's so nice to attend these type of lectures, and hopefully it's will be a continuous process, because generally we are listening to more these kind of topics in this COVID pandemic only. And ultimately, because we have to focus on our academics, and this kind of workshop and seminars will really brush our knowledge with the current scenarios and the latest developments in the field. So thank you so much to the dollar education team and all that people. Thank you, Sandeep sir.
Thank you, Anuj. And this is going to be a continuous series. Like every month, we are going to bring some outstanding scientists to share their journey. And if you know someone whose journey is very inspiring to you, feel free to suggest those names in the feedback form. and we'll follow with them if they can come to our platform and would be willing to talk about their research and career journey. And there's also a nice feedback from Poonam. She said like it's very nice effort for sharing the journeys in scientific community, especially for the early career researcher and future scientist, future PhD students like me. Thank you, Poonam, and we really appreciate for you to coming to this kind of platform and connecting with us. Now, without wasting much of the time, I would like to request Dr. Pamar to talk about his journey and get uh, inspire us more.
Yes, I think we all inspired by Dr. Sandeep Dhanda, and let me actually see it. Screen. Just a minute, I'm sharing the screen. Can you see my screen now?
Yes.
Okay, great. So this career journey session, I want to be like keep it more interactive. So I start with ice breaking question. I ask most of the time whenever actually the same question I ask to people. Let guess them. So what do you think is the most influential person of the last millennium? When you say the millennium in last 1,000 year, any guess? Like yeah, and why you think that person make huge impact in last 1,000 year? What do you think? Who was the person who made the huge impact? So any guess? Okay, let me give some
So
clue.
I think, okay, okay,
Yeah,
Gutenberg is of course the printing, but I feel Gandhi is my choice because he has done a paradigm shift in all the way we had been sticking with wars and to find solutions. So he had a different approach, and this world at this time needs that a lot.
yeah,
So,
definitely. That's
but
really.
Gutenberg, of course, he revolutionized the printing.
Mhm.
So
Yeah,
no two opinions about that. So that's my
yeah.
personal opinion. Yes.
I think all the people like they are great, like what we see in the this picture. But it's just like based on like the uh, in the theme of this presentation, and also I picked this John's Gutenberg here. Definitely, Gandhi made actually huge difference. And so in the Gutenberg case, why they picked the Gutenberg as the most influential person? Because the invention of the printing press, if you see overall, oh, what was the big deal in that? But because of the movable printing press, they were able to uh, print actually a lot of books, and then the knowledge became available to the masses. Before that, it was like only elite class. They had actually all these access to the books, and when it became knowledge became available to everyone, after that you see a lot of the religious revolutions or the scientific discoveries, they all become feasible because the knowledge was available to everyone. And even if you see the modern day the information technology, that's also somewhere come from that actually. uh, so that's why they picked the Jones Gutenberg. And so yeah, this was just for ice breaking case. And let's we can start with the my journey now. I mostly grew up in the western Rajasthan. I'm from the Barmer, Rajasthan. That's Barmer district, and I did my schoolings from the Jodhpur. And in high school, I learned the biology, different botany, zoology, or the physics, chemistry, and all. And after that, I wanted to go into the medical schools, but I couldn't get into the. So I did the bachelor in the biology from Jaina and Vyas University. That was the 3 year degree program. And when I was doing that bachelor degree, so uh, that time biotechnology was very new, and people were talking about this was like buzzword to that biotechnology is the future. And even in my college, they started the biotechnology. There were very few students in that biotechnology uh, degree, but still we find that there's something like the that we can see in the future. Biotechnology will have actually more opportunities. So that's why uh, I try. I actually went to University of Rajasthan and I did my master in biotechnology. I think that was really good time. That first time I find that yeah, all the genetic engineering, and I got so much interest into the molecular biology and the immunology. And so when I was doing the master, I met actually a lot of students in the University of Rajasthan. They were doing some kind of research there. And but I think the most important thing I find is I visited once the JNU when I was in my bachelor. When I was doing the bachelor, I visited JNU. There I meet a lot of people. They were into the research, and they were saying after the PhD you can go out for postdoctoral research and you can get the more exposure. So that kind of I got interest to go. I said I should go and uh, for the higher educations. So when I was doing master, I tried for the different tests, the related to fellowships, the DBT or the CSIR test, and I cleared those tests. And but I was determined that I'll go to the national laboratory somewhere and learn more about where we can really do the have the more infrastructures available for research, and also the people we work with. So I tried a lot of different institutes, and finally I joined the IMTech. It's in the CSR Institute in Chandigarh, the Institute of Microbial Technology. Although the whole institute is related to microbiology, there are few labs they were in the immunology or molecular biology. But what I find really interesting is the Dr. Raghuva's lab. They were working on the bioinformatics, and I still remember after this interview when I went in the lab and see all the people they were working on the computer. It was in the 2000 early 2008, and I was like, "Oh, you guys are doing your whole PhD on the computer?" And they're like, "Yeah, that's possible." So I find that's very interesting, and so that's how the my bioinformatics journey start from there. So in my PhD, we developed a lot of tools there, the database, and learned different machine learning algorithms. And it was, I think, very training was really good, and learned a lot of things from there. And so once I finished my PhD, then I applied for different postdocs, mostly in the United States, because that was where I really wanted to come. And so I got offer from University of Michigan. I joined as a postdoc. That was also the bioinformatics department, uh, computational biology on the bioinformatics department there. So from in my PhD, also in my first postdoc, I was mostly relying on the public database because I was always working in the uh, totally bioinformatics department without much collaborations. We had some collaboration in my Michigan University of Michigan postdoc, but there also they had like they generated some of the data and then say, okay, we want to apply this bioinformatics tool and uh, get some results from there. So it wasn't like the total uh, collaborations where you can see the where you can really apply bioinformatics totally how you want. So and after that also I got some interest in the translational bioinformatics side. I was like, okay, that's fine, we can develop the tools, we can develop the database, we can publish the papers, but what else we can do with the bioinformatics? And so that's why I applied here in Lahore Institutes for Immunology, uh, because the project, they say, okay, this project is funded by Kyowa Kirin, it's a pharmaceutical company, and but it will be totally upon you. Like you can apply any of the bioinformatics tool you want. You can take the lead and come up with some of the totally unbiased data-driven drug targets that Kyovakirin then further check. So that I really find very interesting, and because this whole institute was the immunology institutes, and all that's not that big. That I think 25 or something labs there, but it is very well known all over the world, and I learned a lot of uh, both side, the bioinformatics side and as well as the immunology side from the this institute. And overall, and as I told you that I work both side, the immuno-oncology side and the autoimmunity side, especially the lupus project. We find really interesting drug targets that are being investigated. So this was overall good learning. I spent around 3 years here, and because based on my experience with the Kyovakirin, and I find this is possible. The bioinformatics things are changing every few months. Things are changing. A lot of new advancement you see with the single cell. Now, not only the single cell genomics. Now people are trying the spatial single cell uh, genomics or transcriptomic side. So like we'll get even the higher resolution data every year. We'll see the higher, even actually more resolution of the data, and people are working on all different kind like the high seas data. It's not only the data. So the data is not the important thing. Is like what you can solve with the high C or with the spatial transcriptomic, the way you can look into that data, you cannot look into the other traditional way. So that's the advancement here. But overall it depends on like what you really want to achieve. I don't really uh, see the industry or the academia different. I know in the India it's considered totally different, and but I think I see this is kind of symbiotic relationship, and I think they can coexist very well. And people in US here, they move very freely between the academia and industry based on their interest, like what they. It doesn't matter whether you are in academia or industry, or what really matters is whether you are making any difference there, or like the what impact you are making. I think that should be the criteria. So uh, so after these love institutes, I uh, started looking into the different, like more translational side. I applied into different hospital as well, and in the industries, and I got actually multiple offers from different industries, from all the big pharmas, from the Pfizer, from the J&J, Sanofi, all. So that was an issue. Thing is, like I was more into where I want to go, and I was more interested into the development side. And so as close I can be the development, that was my goal. So in the Amgen, I'm work in the computational biology group. That's the division of clinical biomarker and diagnostic. So where once we start, our work start from the first in human. When the clinical trial start, from there our work also starts. So that's the, I think I find that very interesting, to work on the real data and do some kind of bioinformatics analysis there. And now all the pharma, they are adopting they want more and more uh, informations regarding their molecules, because you see like kind of money they put in the whole clinical trial, the bet is huge. And if bioinformatics or any of these computational analysis, if they can make difference, that's really they are interested in this part. So I think with time it will grow. So that's why I'm working here in the Amazon. So this was the kind of the why from where it started, why I picked that. I wasn't sure. Like for example, when I was doing bachelor, I wasn't sure that what I'll do. I had some idea that I'm interested in the higher educations, but with time it changed, and I kind of kept my option open. Wherever I find my interest, I like, oh, I'll go into this side. So that's my way. I'm not someone who can say, oh, I decided when I was doing that I'll go in this direction. It wasn't like that. I'm always, what's the best available option at this time, and where I find my interest, and where I see the more alignment. So uh, yeah, this is the, like, if you see the summary of my PhD, I won't bore you with this. Like, it's simply like we tried a lot of different machine algorithm methods to solve some kind of biological predictions and publish some of the papers. And so, for example, you can take this tool, the for example RNA-con. That's the classification of different kind of non-coding RNAs. So, given a RNA sequence, can you identify whether it's the tRNA or rRNA or all different kind of uh, RNA, non-coding RNA. So we use like the different because that time I was using the publicly available database. So RFM database we used, and then different classifiers we used to develop this method. It's available in the web form. So it was really good from the learning point of view, like how to really first clean the data, find the data, and then develop the prediction methods and make the web server available. So I think if you are, it depends on what stage you are. If you are in your master or in PhD, uh, I think this is really useful to learn these all the uh, really work with the real data and learn about that. And so this is a whole part of the training. And in my postdoc also, I developed some of the tools, and in this we had the collaboration with two different labs, and also participate in the dream challenges. I think this is the great. I don't know how active the dream challenge is now, but uh, it's similar with the Kaggle. I think the if you are in the bioinformatics student, you should really participate in these competitions. So there, uh, they give the beauty, like they give you the data, and what if they have any biological problem. They give you some of the data. For example, they give you 60 or 70% of their data, and then say, this is the problem we want to predict, and use any machine learning method you want, come up with the solution, and then they test the remaining data. For example, the protein structure prediction also, they used to do all the CASP competition. They still do, but now you see the AlphaFold when it comes from the Google, they beat all all these existing algorithms we have. So uh, yeah, that I encourage you to go and find the kind of the problem you are interested there and work on those. And then they have the different leaderboards, so you can submit your prediction methods there. They will evaluate, and based on you can get the ranking. So I think it's not important to kind of win this competition. All it's just like you learn more and more, and also you also learn that what works in the real life. That's the important thing, because if you go, for example, in the industry, and where you see real time the clinical trials they are going on, they are giving some compound, and then they see measure the efficacy and safety profile, they see some of the efficacy, then also they want to learn why they are seeing this efficacy, or when they see the adverse event, they also want to know why they see the adverse event, whether we can predict in early stage. So there all these are just numbers, like when you publish like the 98% accuracy or 90% accuracy, that's doesn't add there. Like all they want, whether you can in the real life, whether you can predict or not, because it's going to happen in next few months. So I think similar sense you can get from these competitions as well, because that's the kind of, it's not real time, it's still real time, because the data is totally hidden to you. uh, so no way you can over optimize that. If you use any sophisticated, like more over optimized model, you won't see actually in the top ranking, your name in the top ranking. So mostly like people, they with very simple solutions, they see actually those methods works in the real life, by simple linear regression, simple logistic regression, that beat all the other method. A problem is not that difficult. But if you have the huge data, like terabytes of data that you cannot do with these, then definitely you'll go with the deep learning and all. Like, so it all depends on the uh, problem, like what you want to solve. So I think now I feel the bioinformatics or computational biology is really ready for the prime time, and it's not where like uh, biologists, they were seeing these bioinformatics as a kind of helping hand to kind of just verify their hypothesis they have. And uh, but I think it's totally, now you can take the lead, whichever project you are working on, you can take the total lead, can do totally data driven analysis. If you find something, then you can tell biologists that I find something, go and verify. You will have higher chances for to get success from this way. So it's kind of table now turned. So, and this I already presented you the lupus project and the other side of the immuno-oncology projects as well at the La Jolla Institutes. And here I'm currently working at the MGen. So this is all. I think I want to come into the practical guide to the career. And please feel free. The whole purpose when I asked that question for ice-breaking question, so that to get the more involvement. If you have any question in between, please feel free. Yeah. So do you have any questions so far? I just want to, because I want to keep this session where people really find it useful, where they can get what they're really looking for. Okay, so then I can come to actually these some of the things what I feel is the important. So it also depends on where you are now and what you are looking for. So I really find like two different things. One is the skills, and another is the immigration. So I'll talk about these two things. So the skills first, it come to the skills, and skills you learn over the time. For example, in your bachelor you learn, that's very important, like to clear all your basics. And one of the important thing I find is like always your source of information, like what kind of book you are reading. That's the most important thing. Like get the books from the topmost author, not the second top who is the topmost author in that particular field. Read their books. And so, and clear all your basics in your bachelor or master. Try to learn more and more. And in your PhD, also try to work on like different projects. You always like you have your own project, but try to collaborate and learn about different projects as well. Because in the industry, what's really important is your transferable skills. In your PhD or in your postdoc training, if you get so like expert in only one field, then they see that as really kind of very limiting, because uh, things change very quickly. Like if they don't see any kind of the success in one project, they just shut down the project and then they start something else. They have a lot of different programs at the one time, and then they keep changing their priorities. So they want people those have the transferable skills. They can easily move from one project to other. So that will only happen when you have the lot of different skills, or you when you understand actually all sides of, like for example, when you say all sides, for example, you know the epigenetic side, or you know the genomic side, you understand the how the transcriptomic data work, or the proteomic data, or like the network side, or like the all different kind of side. You don't need to be expert. I mean, like they all you need to do little that you are aware about that. It's always you can self learn at that stage. Once you have the proper training, you can always self learn. And there are a lot of tools like available or in the online, so that's not a problem. It's just like uh, you once you have your basics clear and have some kind of the exposure from different kind of skills, so I think that's really good. And the always the core skills as a bioinformatician, what's your core skills, that you should always work on that. So I think the most important core skills is your programming. And it doesn't matter, like the most of the people, they prefer the Python or the R. And you can do any programming, but because for example, if you start working in the group and they prefer one programming language and you are the only one who are doing, for example, the something totally different, so that's sometimes that's can create some issues. But once you have your basic, I think it's very easy to switch from the language, different programming language as well. But at least make sure that you are expert in one language, either it's Python, R, or it can be anything. But yeah, be like expert in that language. And always write like the code where everyone can understand, because in academia really We write code for our own project and only we understand that, and that's not a problem. But especially when a lot of people are working with you, your code need to be very clean that everyone can understand that. And all the basic, the data science tools, explore as many as you can, and the machine learning and AI is like different. Now it's very easy. Earlier you used to actually install all these classifiers separately. Now if you go with the scikit-learn in the Python or in the caret library in the R, all you need actually just change the classifier, you can have. But it doesn't mean then you don't need to learn the basics, like how, for example, when you do the cross validation, how it's really going like in the background, how it do the cross validation. So yeah, always like focus on your basics. If you are in the master or in the PhD work, it depends where, like what stage you are. And the most important thing I find is the biostatistics, that you need to be expert in the uh, all different kind of statistical test. What test you should use in what circumstances, that you should know. Like you should be the person that, oh, this kind of data, if you have this problem, this is the best test you can use. And I find that even though people get a lot of training, very simple, they get like confused with the very simple biostatistics test sometimes. uh, so that's something you can explore more. And the genomics tools and technology, because it will keep evolving. So for example, if you looking for PhD, and if you are like an early stage of your PhD, and think about like when you finished your PhD after 5 years, there'll be totally new technology. And so, and because it happens sometime, because when you work in the lab and uh, people prefer like one particular technology and that coming from like last few years. So make sure that you are working on the cutting edge technologies, or like somewhere you have the intersection or in the collaboration that you know about that. So uh, always, yeah, keep up to date, like work on the latest technology you have available. So these are, I think, the all the skills you can build. And after that, like once you have the skills, how to reflect that skills proper, like in your resume or in your cover letter. And so that's also like, I think there are a lot of online where you can learn about this, not the uh, something actually I can go and the explain all. But yeah, definitely you need in your resume, what the important thing is, your what you learn from every project you work, what you learn from there and what problem you solve, that you should always mention. And in the cover letter, even if it's like one paragraph, you can always say like, oh, this was the project and we wanted to solve this problem and this was the outcome. So just like brief summary, so that people know that uh, what you did actually. Otherwise there, if, a lot of time, if you see the resume, people just mention about the problem, but they don't give actually what they solved there, actually how they solved that, or what they achieved out of that, people don't mention. So yeah, this is the very like practical guide, that's what I feel can help you. And the networking always good, and you can, it's easy through the LinkedIn, you can approach the people you are interested into and keep building your network. uh, it's very easy now, I think, through the LinkedIn you can approach anyone and you can learn about that. And most of the time people, they are very helpful, so it's easy to get information. And the location, because I think the physical, if you see now, getting more and more blur, the you don't need to be in that locations, but still it is important, I find. Like if you are close to where most of the opportunities are, you will have higher chances. So but yeah, that's something you can keep in mind. So this is like related to the skills. And another, this only apply, the immigration part only apply if you are interested in the US. But I think in general, like if you want to stay in the India, then it doesn't matter. But if you want to go anywhere, I think it's important to know whether the country you are interested in, they are like uh, they support the immigrations, how much they support, because over the time it will really uh, it will be important for you, because if it takes too long just to get the immigration right, then you sometimes you lose the opportunities because of that, not because of like whether you are capable or not. So that's something in also need to take. Like if you are in the master in the PhD in the India, definitely you should think right from you can start thinking now, so that because this takes time also. Mostly like they sponsor the J1 visa if you come for the postdoc. So that's the J1 visa. But on the J1 visa, you can normally not work on the industry here. Hard to move from the J1. So normally people spend some time on J1 and then they move to the H1B visa or O1 visa. Industry can sponsor the H1 visa or H1B or the O1 visa. Uh, but normally they prefer people those who have the green card. But it also depends. Like they can sponsor O1 or H1. So it's all depend on the requirement. So in the green card, there's different categories: EB1 or EB1 or EB2. So EB1 is the top priority categories. This also further into like EB1A or EB1B. EB1A where you self-sponsored, you apply yourself, and EB1B where your employer apply for you. But overall, the EB1, the priority date is current. So it means like once you have your EB1 uh, application submitted, you can within few like year or you can get your green card. But only three important things you need to work on right now, because that's what I'm saying. Like even if you are in the PhD now in India, or you can think about these three categories. What they want first, they want is your original contribution to the science, like your publications, what you contributed to the science. So that's something you already do in your PhDs. Or and another they want is the contribution in other people's work, like whether your work used by other people's, like through the citations. So you can always keep track of your citations, like who are the people you're citing your work, and if you can get recommendation letter from them, that will be useful in your applications. And third is whether you have judged other people's work. It takes time. So here it means like whether you reviewed the manuscripts. So it takes time, but you can always approach the editors from different journals and from your PIE. And but ideally the email should come under your name. So these are the few like things where you can work on. So once you have these things ready, then you can apply for green card. But why I'm saying you this, it's maybe for some people like it doesn't look that important, but I'm saying you because once you are late, like if you don't work on this and suddenly you realize, oh, you need all these things, then it takes a lot of time. You cannot build a review like all like you cannot have all these like in very short time. So yeah, just keep this in the mind. I think yeah, it will be helpful some for somewhere. So this was from my guide, and yeah, please feel free if you have any question. I'll be happy to answer.
Can I comment? If you
Yes.
permit me?
Yes, sure.
Minutes? Yeah, it was truly an incredible journey. I congratulate you the way you have risen in your career, starting from Jodhpur, a very not a very modern place, but yes, you have risen to this level and reached the best industry. Amgen is the top in the world. It's really an incredible and a very inspiring journey. I'm really impressed. Two, 3 points I would like to make since it's an interactive session and it is for motivating the students also.
Mm-hmm.
It's very important as you emphasized on the fundamentals because
Yes.
I teach bachelor's and master's
Yes.
and teaching is my passion. So I had also lots of offers, but I thought to remain in teaching because that was my forte. And I make it a point that I teach students in a way that the foundations are very, very strong because ultimately that will matter in their career and how far they will go in the career. So I give a
Definitely.
lot of time on
Yes.
strengthening the fundamentals. And basically, I'm a virologist, and many
Oh.
students have done proud, and they are one of the top virologists in the world right now.
Oh,
Sanjay
that's great.
Fogart is one. He's with Sanofi, and he
Oh,
worked extensively on HIV.
mhm.
So he's one of the best students.
That's
Then
great.
you talked about IMTech.
Yes.
Two of my students, if I can recall, the first batch was Deepak Nilani. He worked with Dr. Sahani on
Yes.
streptokinase.
Yes. Yes.
That was at IMTech, and then other person was Shudipta Saha. He worked with Dr. Raghava,
Yes. Yes.
and
He was our senior.
he's now at the Bose Institute. So
Yes.
these two people, offhand, I can just recollect that they entered into bioinformatics
Yes.
as such.
Mhm.
So this is very important. What are your choices, and what exactly the choices? Always make that which you are passionate about.
Yeah.
Then,
Definitely.
as far as you talked about the self learning, see, I'm a
Yes.
virologist, and it's only been 3 years that
Mhm.
I taught myself bioinformatics because
Okay.
I saw that it has so much of potential, and
Uh-huh.
without bioinformatics, you can't survive these days. So it was a big challenge, and nobody wanted to teach bioinformatics in my department. So I took the challenge at this age of 60, and I took up that challenge and started leading the online courses. And that's why I'm here, trying to
Oh,
learn
that's
more
really
about
incredible.
bioinformatics.
That's
So
great.
I mean, but that's interesting.
Mhm.
Bioinformatics power, as you can see, the
Yes.
synthetic biology and bioinformatics is
Mhm.
only from the in silico that we made the messenger RNA of
Mhm.
SARS-CoV-2, and we
Yes.
designed a vaccine, if you
Yes.
know this, right?
Mhm.
So
Yes.
that is the power of bioinformatics, in silico power, and the synthetic
Yeah.
biology. And the most two successful vaccines, of course, are the Moderna and the Pfizer. As you
Yes.
can see, these are the best vaccines. So that is the power of bioinformatics. So I think I encourage students to take this course very seriously and work on this. So these were some of the points that I just wanted. I don't want to take much of your time.
No, that was
So
really incredible. Yeah, that was really
yeah,
inspiring.
yeah. Even I was born in Bharatpur, so I'm really
Oh,
proud of
okay.
Rajasthan.
Great. Bitter. Yeah.
That state I really have a lot of regard and respect for the people, because those people, they really have a character and they never show off.
Mhm.
Like in Delhi, I live in Delhi, and I find people, they're not knowing anything, but they show off so much. But people from Rajasthan, they know so much, but they never show off. With that parting note, I will end, and thank you so much.
Thank you. Yeah, I encourage actually students if they have any question and they want to know anything. Yeah, please feel free.
There is a question in the chat from the Poonam. Should I read that one?
Yeah, definitely.
Yeah, so she's asking, sir, I had 2 year research experience in wet lab. Then I had few year gap due to health reasons.
Mhm.
Now I want to return to research as interest always stayed with me. As a kind, I have to start over. Can you please suggest me a career in biotics? What is better, master in computational biology, biotics, or PhD with focus on computational biology?
The master, what
Yeah,
stage is the
she's
master?
kind of, if I'm trying to interpret, she's kind of proceed career into biotics. So should she start with MSc in biotics, or can she continue the same thing after a PhD with focus on computational biology?
Oh, so master versus the PhD. So it all depends on like your uh, whether you can spend that like PhD normally it's uh, like you need to commit like more years. And so if you have that time that you can spend like for PhD, then definitely I would suggest uh, go into the PhD because that would be the you'll learn more and you'll have like your own project where you can basically explore more. I think in the bioinformatics, but it all I think it totally depend on your personal. If you think like you want like within like 2 year or something you want to go for the job, then I think master is the best option. And also I would say like you can approach different industry also in the India wherever you are actually get some internship from there. That's I think even like if you're in the PhD you should always go. That's very common in the US that I don't see actually in India. People here in their PhD they will always go for internship, or they in the master they will go for internship. They spend some time in the industry and most of the time you see like they then join that company as a full-time employee. That's the trend. So that's also you can something you can, but yeah, it totally depend on it. It depend on your situation. If you want to do master and just want to have job after the master, or you want to spend like more time for PhD. If you can spend more time, then definitely go for PhD. And there's so many like good bioinformatics labs in India, so that will be worth, I think. Yes. Any other question? And also, I think like you always have options to do master, or for master normally like it is little like expensive doing outside, but for PhD you can always apply outside also in the Europe or in the there's so many good laboratories as I told you like in the India as well. So definitely, but if you are interested you can always apply outside as well, like in the Europe, in the US. In thing is in US normally it takes long time for PhD. In Europe people get like the relatively they get actually early PhD. So Europe also good option. Germany or UK, a lot of from only thing is like there you get the other experience as well in different culture. You know a lot of you learn a lot of other things as well. So that also you can consider. Yeah. Any other question people have?
Yeah, we encourage participants to ask questions because that's the ultimate motive of this series. Yeah, just to summarize what Dr. Pamar was saying, like even you are looking for a long-term career in bioinformatics. So these two things that are really important here. The one is uh, transferable skills. Like you should work on these things, how you can learn it quickly, and how you can go through and keep adding more skills over there. The next is to work around the immigration part. So we do not want the talented people to stuck because of immigration uh, issues. So that's why if you are aware in early stage, many of the things you can figure out. And those kind of the two lessons that I have always learned from Dr. Pamar, that he is kind of a mentor for me. And he always encouraged like these are the things you should really work on if you want to continue your career here, you want to continue career there. So he is very open about that, and that's why I invite a participant to ask their questions. So there's also, oh, by this kind of encouragement, we got a question. Let me read out that.
Yeah, sure.
The question is, sir, I have a master. The question is asked by Roja. So, sir, I have done my master's in bioinformatics and dissertation in plant transcriptomics study.
Mhm.
If I will go to medical research in microbiome study for metagenomics and metatranscriptomics, how will be the future in this field? And there's also a follow-up question. Also want to know what is the transcriptomic and metatranscriptomics.
Okay, so she's a master student. So I think like these are all if you see the plant transcriptomic or the uh, simple transcriptomic or meta transcriptomic, where you have the kind of the data from different bacterial or like different microbes all together. So I think these are all the distinction we make. uh, I think at this stage, if you are in the master or even in your PhD, you should not make distinction that you are like in the plant bioinformatics, or like you are in the I think overall the skills you learn from the bioinformatics, that you can apply everywhere. So that should be the goal. Uh, Unless you want to be expert of one particular field, that's like you so determined that I want to be top most immunologist or something, then that's a different story. Then don't take my advice in that case. But I'm just talking about like in career point of view, like when you where you want to have more options. So definitely I see the future of the microbiome and the med, because it lot of new evidence they coming from that it affects your overall, for example, the immune status as well, or like the all the different kind of diseases, autoimmune diseases, or even in the cancer immunotherapy, they see the role of the microbiome. So definitely that's the uh, field. It's emerging field, and you can work on that in your master or definitely. But what I'm saying, don't restrict to only microbiome. Why you should restrict to only microbiome? uh, Go for the multiomics, like where you do all kind of like genomics, like you should know like how these variant works, or like different kind of genomics tools works, or how you can use that, or like the transcriptomic, how to do that proteomics data, or like the microbiome also. They are all the you should know like all different kind of technology and like the data, and work on that. But yeah, definitely for your project, if you find so much interest in the microbiome, that's totally fine. But what I'm saying, like keep your options little open. And uh, when I'm saying that, it doesn't mean like the microbiome has the huge opportunity. It's if you do like anything with master and like project related to microbiome, it has the huge opportunity. So please go with that. But over the time, just try to learn like the all different kind of omics data, like how to interpret that data. Because I've seen actually people, like for example, they are kind of expert of the transcriptomic data, but when you see like from the genomics data how to interpret that, they don't know much. But if you, as a bioinformatician, I think you should know everything, all different kind of omics data. You should know in the where at least like the tools you are aware about, and if someone give you that data, you should be able to interpret actually. So that's the skills like you can build in your master on your in your PhD without restricting yourself. Because we can understand like in the experiment when you are experimental biologist, that's a different case where they have like for example this particular protein we want to characterize, and there it totally makes sense why they go in that. But as a bioinformatician, where you have a lot of options to play with all different kind of data, and especially important thing is like uh, what is really important is how you integrate different kind of data. Like if the similar like microbiome data you're working on, that's fine. But then if you have any other data, how to integrate and get more signals from your microbiome data, that's the important thing. Like how to integrate the different multiomics and get sense out of that. Because for example, I give you one example, the lot of industry they are working on like the early detection of the cancer from the circulatory tumor DNA. So that's actually something the fragmented DNA from the tumor cells gets into the bloodstream, and from there, if you can detect the early stage. The problem is like if you see the most of the DNA coming from the healthy cells, very tiny amount is from the tumor cells. So there's a problem of signal noise ratio. Signal is very weak. So what algorithm you can use, how to basically enhance that signal, and then basically predict reliably. So there's so many problems like this in the open problems in the bioinformatics, like where. So now this like now you can, if any of the other omics that can enhance that signal, that's useful. So what I see actually is go after the problem. Always use the cutting edge technology. That's because that will help you, and always go with the big problem in whatever field you are. Like, what is the big problem there? Solve with the cutting edge technology. That should be the goal. There are a lot of the good labs like Rob Knight's in the UCSD, and like there are a lot of big labs in the microbiome space. So you can learn from like what they are publishing, what tools they are developing. So you know what's going on in your field.
So are there any other questions that you want to ask? It could be related to your courses, it could be related to your immigration, it could be related to your project. So that's the ultimate kind of purpose of this series.
And I really appreciate Sandeep and the Dollar Educations for real this series. I think yeah, it will be really helpful for people, and I think that's something I feel like when I was in master or PhD, and if I had chance to meet, and I think yeah, I would definitely take some advantage of that. That's something you should always take advantage.
Is Poonam is asking about internship opportunity in India, and I would again kind of advertise dollar education in here to kind of stay tuned about the many updates that we are pushing through the dollar education. And other Dr. Pamar can add if he has more experience on this.
Yeah, so definitely like the internship in the India, the internship you can learn like from the Sandeep. He knows more about that, and I think like mostly through your professional networking. If you know people like in the industry, then or this advertise also. But normally, if you know, you can ask them like if you have any internship opportunity available, and so they I think they publish in LinkedIn as well if they have the opportunity available. But mostly, I think it comes through your networking, and also like based on your interest, you can figure out what are the industries there. Uh, and for example, in the bioinformatics, there are a lot of like industries now. You see, they are totally bioinformatics based. So if you are a bioinformatics student, you can prefer those industries. Doesn't matter even like if they are relatively small industries, because you learn more uh, about your field there, where all the people like they kind of focused like themes on the computational biology, but you can target the industries where you are interested in. But yeah, definitely reach out. For example, you can directly find like in the LinkedIn uh, people from that industry, and like mostly like the HR people from those. You can ask them also. If you don't find any other people, you can always ask HR people that if there is any internship available, if they are aware about any of the division where they, or like sometimes they say we'll have available in some like in 2-3 months or something. Then also you can keep in contact. So it's all depends on like it will be always depend on your efforts, uh, uh, how much effort you are making, and like where you really want to go for internship. So but that will definitely help. Like I really encourage all the, because that I haven't seen in when I was doing PhD, uh, because spending all of your PhD in one lab, I think that is good. But yeah, you should spend some time in internship as well. That will give you really edge over other lot of other things. Like so, you will learn more. Great. So, is there any other question? I see some of the.
All right, if there are no more questions, so I think Sweta can now take over the session ending.
Thank you so much, all. Have a good weekend.
Thanks to you, sir. And yeah, she's gonna have some announcement, I guess. Yeah.
Thank you so much, sir. Thank you so much, everyone. Thank you so much, sir, for such an inspiring talk and session. I hope participants got a lot to learn from here. On behalf of Dollar Education, we are launching our next paid workshop on bioinformatics for beginners. In this, various different topics will be covered. Introduction to the bioinformatics, nucleotide sequence database, protein sequence database, BLAST, multiple sequence alignment, phylogenetic analysis, primer designing, and network analysis in cytoscape genome browsers. Register yourself at www.dollareducation.organization. Thank you, everyone. See you all with new session. Till then, enjoy learning. Thank you so much, sir.
Thank you.
Thank you all the participants, and thank you, sir.
Thank you. Okay. Bye.
Thank you. Thank you so much, everybody. Thank you.
Bye.